आओ नमन करे।
1987 में मैत्रेयी कॉलेज नेताजी नगर की स्कूल बिल्डिंग से चाणक्यपुर स्थित इस आकर्षक भव्य बिल्डिंग में पूर्ण रूप से शिफ्ट हो गया। प्रिंसिपल डॉक्टर सीता श्रीवास्तवा का अत्यधिक महत्वपूर्ण योगदान, जिसके लिए मैत्रेयी समुदाय सदैव उनका आभारी रहेगा। अपने कार्यकाल के दौरान उन्होंने कॉलेज की वर्तमान बिल्डिंग के हर एक विंग का विशेष रुचि लेकर निर्माण करवाया। इन्हीं बीते पलों और मीठी यादों को एक खूबसूरत अंदाज में सहेज कर बैठा है मैत्रेयी कॉलेज का ये आकर्षक परिसर। 1989 से प्रिंसिपल श्रीमती स्वदेश भल्ला की सकारात्मक सूझबूझ से कॉलेज बहुपक्षीय विकास की ओर अग्रसर हुआ।
During my tenure, honors in physics, chemistry, zoology, botany, sociology and in computer science, they were introduced. So during that time, automatically we saw to it that all our labs, because all the faculty was to be appointed in the science departments, all the labs were well equipped so that the students could do their best in this, and we succeeded in it. During that year only, we started with our cultural festival, which became a every year event, and even today we are following that tradition. We started Rhapsody, then we succeeded also in constructing a multi-purpose hall so that this could help the students of the college. Lawns were there, our Malis worked really hard to make it a green campus. Not only that, it was recognized by the university, and year after year in lawns and garden we used to get the prizes. This is what the situation turned out to be. As far as teachers are concerned, their achievements also are a lot. 5 teachers of the college were given positions of principals in other different colleges of the University of Delhi, they being late Dr. Mrs. Savita Datta, then Meera Ramachandran, Vijay Lakshmi Pandit, Bharti Sarkar, Premrata. Dr. Shashi Kumar became the Vice Chancellor. Amarjeet Kaur from the Physics Department, she was awarded the Young Scientist Award by the Government of India. Dr. Shashi Tiwari, Chairman of WAVES, an international forum which was created by her. Dr. Mala Kapoor,
Dr. Shiva has joined us. Welcome you, sir.
Thank you.
So we are going to begin shortly.
Sure.
Am I audible to all?
Yes, ma'am, you are audible.
Okay. A very good morning to eminent scholars and esteemed guests present here today. Here, I, Dr. Lata Vodwal, Assistant Professor, Maitreyi College, welcome you all in technical session 3rd on the second day of the International E-Conference organized by the Department of Chemistry, Maitreyi College, University of Delhi. Today's the session coordinator for this technical session is Dr. Manju Mehta. Dr. Manju Mehta is an associate professor in the Department of Chemistry, Maitreyi College. She has more than 27 years of teaching experience at the undergraduate level. She did her BSc in chemistry in year 1987, MSc in year 1989 with the specialization of organic chemistry, and she earned her PhD in year 1993 from the Department of Chemistry, University of Delhi. She is the recipient of Merit Certificate awarded by CBSE India for being among the top 1% student in senior school examination. And she also received undergraduate Science Merit Award from the University of Delhi. She has published many research articles with high impact in several reputed national and international journals. She has been extensively involved in writing, publishing, and editing e-content for undergraduate students. Besides her research publication, she is the co-author of book entitled Organic Chemistry, published by PHI Learning Private Limited, New Delhi. Her authored book is studied extensively by undergraduate students throughout India and is cited as one of the reference books by various universities. First edition of her book was published in January 2005, and till date the book has been printed 11 times from the year of publication. Besides chemistry, her other area of interest is psychology. She did online certificate courses through SWAM, NPTEL, and awarded with elite and silver certificates. I welcome you, ma'am. For this session, we have two invited speakers. Dr. Shiva Shankar Panda from Augusta University, USA, and Dr. Upendra Kumar Singh from La Jolla Institute, Immunology, USA. Now, I request Dr. Prajalit Shikha to welcome and introduce our first invited speaker for this session. Over to you, Dr. Prajalit.
Thank you, Lata ma'am. A very good morning, everyone. I, Dr. Prajwalit Shikha, Assistant Professor, Maitreyi College, University of Delhi, welcome you all once again on the first session on second day of this international conference. It's my great pleasure to introduce our speaker, Dr. Shiva Shankar Panda, who is going to deliver his lecture on rational strategies for the development of potential drug candidates against SARS-CoV-2. Dr. Shiva has done his M.Pharma in pharmaceutical chemistry from MCOPS Manipal University in 2005 and did his PhD in medicinal chemistry from University of Delhi in year 2010. Sir then moved to University of Florida, Gainesville, USA to carry his postdoctoral research where he worked along many senior professors. Sir has also worked as senior scientist in Sigma Aldrich, Miamisburg, Ohio, USA. Currently, Sir is working as an associate professor in the Department of Chemistry and Physics, Augusta University, USA. Sir has vast teaching experience and has mentored many undergraduate students. Sir has taught various UG and PG courses and supervised many graduate students and postdoctoral research scholars. Sir has got many scholarships and grants from various agencies, Cultural and Educational Bureau Scholarship Award, Educational Innovation Fund, Pilot Study Research Program Award in collaboration with Cancer Center, Center for Undergraduate Research and Scholarship Summer Scholar Award, Translational Research Program Award, Center for Undergraduate Research and Scholarship Award, to name a few. Sir has got Excellence in Research Award from Augusta University in 2018. Sir has also been the member of American Chemical Society since 2014. He has various national, international, and local collaborations and has published a number of manuscripts and peer-reviewed articles. Sir has been the part of various committees at Augusta University for faculty development, design and development of courses, and career development. Dr. Shiva is in the editorial board of various journals and is also the reviewer of a number of journals. Sir has published more than 90 research papers in various reputed and peer-reviewed journals. Sir has also published 3 book chapters and has got 3 patents on his name. Sir has been invited for lectures in various workshops and conferences and has presented papers in more than 30 conferences. Dr. Shiva, a renowned personality in the field of medicinal chemistry and drug designing, is present between us today, and I'm sure that we are going to get enriched by his knowledge and experiences. On behalf of all the participants here, I welcome you, sir, once again in this conference. Thank you, and over to you, sir.
Thank you so much for the nice introduction. Today we are going to talk about the rational strategies for the development of potential drug candidate for COVID-19, where we are facing a pandemic at this current time. So when we're talking about this COVID-19, we are talking about the problem, the pandemic we are facing at this point. But before we are talking a little bit more about the drug development, let's talk about a little bit about this coronavirus. And I'm sure we do not need any introduction for this nasty virus, but still I would like to mention the numbers where we are at this point. So more than 290 million people are got infected by this coronavirus. And among them, around 34 millions from India got infected by this virus. So more than 4.55 millions got death because of this coronavirus. However, among these 4.55 million deaths, around 450,000 people got died because of this coronavirus only from India. Okay, so now let's talk about a little bit more about the vaccinations. So since we are struggling to get a treatment for this COVID-19, we are exploring various possibilities to how to get rid of this situation. So meanwhile, there are several vaccines got emergency approved, and some of them are effective to have the control about these infections. When we are talking about the vaccination rate at this point, this world is around 46.3% vaccinated. Now you can see from this chart where we stand, but still the numbers are going up and up and up, and people are still struggling to survive. But the more important thing here is in the period of last 1 year, you can see more than 6 variants came up. And because of the new variants, the vaccinations what we have is getting less effective. And this number of vaccinations are increasing, but the effectiveness of these vaccinations are also keep on going down because of these new variants where we are seeing. So now the first one, the variant came is alpha, then beta, then gamma, delta, lambda. Now currently we are listening a new variant about the mu. Because of the different variants, the existing drugs what we are keep on trying to get treated for this COVID vaccine, COVID infections, but still we are struggling to get complete cure, and because of that the death numbers are still going up and up. When we're talking about this, let's talk about the drug discovery process for this COVID-19. So we do not have a definite drug candidate for this virus yet. But still, when we're talking about the discovery of drug COVID-19, then the things comes why it is taking so much of time, why it is so difficult. And when we're talking about this process of the drug development, and we are facing a lot of difficulties to getting a better drug candidate to treat this coronavirus infections. And then we need to understand the drug development process. When we are going through this drug development process, I believe we all know the process of developing a new drug for any type of disease or any type of problems. So to get a drug for any problems, it takes around 14 to 15 years. And it also costs a lot of money to find a drug candidate. Now the problem is, do we have this much of time to wait to get a drug for this COVID-19? I guess everyone is saying no, we cannot wait 10 to 15 years. So if we cannot wait 10 to 15 years, what are the alternative ways? How we can achieve the target, how we can achieve, or how we can get a potential drug to treat these infections. Even though we are having various vaccines throughout this world, but still we need a drug for any kind of adverse situations. So for that, we need to understand the drug cycle process, and then we need to figure it out where this 15 years process can be squeezed, because in this total of 15 years, as we know that we cannot wait for this 15 years, we need to figure it out where are the areas we can squeeze. For example, for this development of vaccine also takes a lot of time, but because of the need of this world, the process of this development of the vaccine, some steps got squeezed, and for that, and we got emergency vaccines, and fortunately most of the vaccines are working, and we are not getting so much of adverse effect. But still, because of the new variants, this vaccination effectives are going down, and some people are getting hesitant to take the vaccine because they are still scared of the consequences because of this vaccine. So now think about that alternative drug candidate we should have to treat any type of adverse situation, any type of infection, because still we are having a lot of deaths. As I did mention to you, more than 450,000 people got died because of this COVID-19 only in India. And throughout the world, it is touching 5 million. Think about it, the number is so scary. So we need to little bit more careful while in the drug development process, and we should not go anything wrong, or we should not do anything longer way to achieve this goal. So for that, we need to understand better the drug development cycle, the drug discovery cycle. In this drug discovery cycle, one of the more important area is the lead compound. So if we see the process of 10 to 15 years, the identifying a lead compound takes around 3 to 4 years. So if we can achieve this lead compound in a minimum amount of time, then from that 15 years we can take off that 4 years, then the process of drug development will be 10 years, and then so on. We need to keep on finding where the steps we can squeeze, and then within 2-3 years or 4 years we can have a drug for this coronavirus. Now when we're talking about the lead compound, how we can squeeze, then we need to understand the source of lead compounds. The source of lead compound means the natural drugs, where we can use different natural products for this treatment, or we can find the synthetic world. That means there are a lot of synthetic molecules are available throughout this world. Every research institution, academic places, and industries, they have a lot of molecules, and they are keep on working. Now let's explore those molecules for this virus and see if any of them are working against this virus. Then other 3-4 years of time can be saved. In addition to that, the virtual world, that means the computational programming, the computer aided drug designing, could be act as an additional tool or supporting tool to achieve our target. So because sometimes the computational drug designing process helps help us to avoid the drugs where we are working, which could be a problem maker. That means if we are working in a molecule which could thinking that it could be a good drug candidate, but because of some other factors which we are not considering, for example, if we are developing a drug and say, oh no, this molecule is so much active against this coronavirus, but when a patient is consuming, then because of some restrictions, for example, if someone is having a kidney problem, if someone is having heart problem, if someone is having liver problem, they cannot take the drug. If someone is pregnant, they cannot take the drug because of some limitations. Or if someone is taking orally, the drug is not absorbed. So because in the drug development process, effectiveness potency is not only one factor. There are so many factors we need to consider, and that only possible when the drug is ready and we can do some animal studies or some kind of in vivo studies. But for that also it takes a lot of time, and once you get a molecule and do the in vivo studies and then you figured out, oh no, this molecule is not working, then all these times what we have put to come to that point is going to be wasted. So to avoid those kind of problems, the possible problems, the computer aided drug designing process, the computer tools or the computer programming helps to reduce those kind of issues. Okay, so now, as I did mention to you, in the synthetic world, there are a lot of molecules. Probably in your institution, in your college also, there are a lot of people are working in a different research project. They have a lot of compounds. And we don't know, one of that could be a good candidate for this coronavirus. But the problem is we have huge number of molecules. How we will know which molecule is going to good fit for this problem? So for that, we cannot test it because it will take ages to test all these molecules ever. Because if you look at the number of molecules, it's a huge. And then for finding a right molecule for the right target is one of the biggest challenging thing. So how we can achieve that? So for that, we need to use this computational programming to solve this problem or to ease this problem process. So for that, there are usually two basic ways to deal with. One is the ligand-based drug designing, another one is the structure-based drug design. What is the difference between the ligand-based and the structure-based drug designing? Ligand-based drug designing, where we do not know the target. For example, when this COVID problem came in 2019, December of 2019, technically we see the real problem in March of 2020. And when we see this problem, people start getting infection and people get died, and then doctors are helpless because they do not know how to treat. And at that time, the researchers, scientists, they do not know how this virus looks like, how this virus is, which protein is having, what is the properties of the virus. At that time, the drug development approach people were using, or to which drug can be used, is they were talking about the ligand based. That means try what are the molecules which are showing some kind of activity against this coronavirus and try to use the similar types of drugs or similar types of molecules for that problem. So in the next part of the drug development process is the structure based, because later in 2020, researchers and scientists identified the coronavirus fully, that they identified the protein, they identified the receptor, then they identified how to deal with this protein. Then the structure-based drug design come into the picture. In the structure-based drug designing, once we know the structure of the protein, we can design a molecule how to deal with that protein. So coming a little bit in details in the structure-based drug designing, which we, in other word, we can say in computational world, that is the molecular docking. In this process, what we know is we know the structure of the virus now. Now we know the structure of the virus. Let's do the docking by using the computational programming so that the process of the drug development will be faster. So in that case, let's find out what are the possible drugs would we assume that these molecules could be a good candidate for this virus. So once we do the docking studies, in the docking studies, there are two steps. One is the sampling, another one is the scoring. What is sampling and what is scoring means? Sampling means we need to identify the core structure and some because when we're talking about the protein, and as we know, the protein is made up of amino acid. Hundreds of amino acids are there, and which amino acids are playing important role, which amino acids are going to interact with the drug, we do not know very well. So we need to find out, we need to choose some of the possible amino acid which has that ability to interact. That means a hydrogen bond donor interaction, a hydrogen bond acceptor interaction, or hydrophobic interactions. So this is the step of the sampling. Once we identify these are the specific amino acid, or this is the part, and this 3 or 4 or 5 amino acids are playing important role in the active site, and then it is coming to the scoring. In the scoring process, your molecule, which we assume that it could be a good candidate for the viral infection, there this molecule is interacting with those amino acids or those active sites or not, and depending on how well it is interacting, a score will come up, and that score will let you know your molecule could be effective or not. We do not need to make that molecule, we do not need to really test it. If your molecule, whatever you design, whatever you are drawing in your computer, if the molecule is showing effectiveness, then it is giving a kind of a signal, let's go ahead and make this molecule. If it is not showing that property, then probably you can skip from that step. Okay, now the next step is the ligand-based drug designing. When you're talking about the ligand-based drug designing, I always give an example of morphine. As we all know, this morphine is an opioid, it's an opioid drug. And this morphine was isolated from this uh, opioid poppies, and then morphine was identified as a potential analgesic drug. That means it is having a very good pain relieving property. Till now, people are using is this molecule for the severe pain. Then later on, there are two more components were isolated from that opioid, codeine and heroin. And if you look at these structures, the structures look similar. And based on the structure, when the structures molecules were identified, isolated and identified, and they saw the structures, as soon as the researcher, the scientists saw the structure, they said, oh, it looks like morphine. This molecule should be effective for analgesic or the pain management. And fortunately, this codeine and heroin, they both are very good analgesic, even though all three are having drug addiction properties, but all three molecules are showing so much of better pain management properties. So now the question is, just looking these structures is good enough to say, okay, these molecules are having showing same properties. Now if you look at these three pictures, and the first this is banana, this is a soccer ball, and this is a watermelon. If you look at the shape, structure, size, color, and all, then probably we will say, okay, this watermelon and this soccer ball looks alike, but their properties are completely different. However, if you look at this banana and this watermelon, see, they completely look different in terms of shape, in terms of color and all, but the properties having similarities. They both are fruits. So now the question is, just looking the structure or just looking the color is good enough to be considered as a similarities in terms of the biological properties, in terms of the biological outcomes? No, probably not. So we need to consider a little bit more than that. So that for that, this 2D QSR approach was used. In 2D QSR approach, where they consider the physiochemical properties of the molecule. Then what is the physiochemical properties means? That means when you look at the molecule, don't just look at the structure. We need to look the little bit more than that. What are the things we need to look? We need to look the hydrophobicity of the molecule. We need to look at what are the substituents, what are the electronics properties, what is the steric properties, is there any electron withdrawing group, is there any electron releasing group, or is there any other kind of effect, is there any electron electronegative atom is there. So many things we need to consider. So it is very difficult just looking at the structure and considering all these properties, but we don't need to worry much about that, because this computational programming helps a lot to do this job. If you have two molecules, if you just upload these two molecules in a QSAR computational program, the program will identify what are the similarities between these two molecules, and then with the life will be much more easier. So what happens is you need to collect all these molecules which are showing similar types of activities. For example, if you're looking for, let's say you're looking for a cancer properties, if you see in the literature so much of diverse structures, starting from Taxol to 5-fluorouracil to so many different types of molecules are showing anti-cancer properties. Now, how can you say that all these molecules are having similar properties? If you look at these structures, they look very different. However, when you use this QSR programming, this QSR programming, this computational programming will identify what are the common parameters among these molecules are having, and because of those parameters, the molecules are showing a specific type of activity. Now, when you are going to design a molecule, and then even you upload in this program without making the molecule in your laboratory, just design the molecule and upload in this computational program, the computational program will say your molecule is having all these required parameter to show this anti-cancer property or not. If your computational program will say, well, your molecule is not having all the required components which you are looking for a anti-cancer properties, then probably you will need to think a little bit differently to design the molecule, rather than making the molecule and spending a month or years, and then you ended up with saying, oh, your molecule is not active. So now our life is much more easier, much more simple because of this computational program. But once you come up with a molecule which is this computational programming is saying that, oh yeah, your molecule is having all these required parameters to be active against the cancer cell lines or cancer properties, then probably you need to go ahead into the lab and make that molecule and do the screening. But now you can say that, you can ask me, oh, is it possible to make drug for any kind of problem? Well, this virtual world is going to give a kind of a guidance. There is not 100% sure that this, if the computational program is saying that this molecule is going to be active against this problem and you are going to end up with a drug candidate. It is not 100% sure, because drug development process is not relying on one component. There are so many factors. However, the possibilities of success is higher if we are using these tools. If we do not use these tools, then the possibilities are very less. Okay, so now moving forward, there is another approach called the pharmacophore approach. And since we are talking about this morphine, and as we are discussed that morphine is having analgesic properties, that means it has that ability to reduce the pain, a severe pain. So if you're talking about going a little bit more, the pharmacophore properties. Now, if you look at the structure is so complex. If you are going to ask to make this molecule in your laboratory, then you need to think twice. Oh my God, it looks so difficult to make it because the structure is so complex. So now let's figure it out. What are the active components, or what are the groups, or what are the part of the molecule is really needed for this biological property, for this analgesic properties? If we can identify that, then our life will be much more easier. So now, how we can do that? So for example, when you are going to make this molecule, take off this OH group, then do the studies. And then if your activity is going up or down, then you can identify, or you can sure, okay, this group is useful or not. For example, when they take off this OH group from this molecule and did the activity and did the analgesic property studies, they did not find any change in the analgesic properties. That indicates this OH groups is not playing any important role. Similarly, this ring, this 5-membered ring here with the oxygen, is playing any important role? No. So what are the component? What are the structural feature of this molecule is playing important role to show this analgesic properties. You can see the red highlighted part. So if this is the only part we need, so why we need to worry about to making this complex structure? So once this component was identified, people synthesized the simple molecule out of it, which is called the metazocine, which is a commercially available analgesic drug. And now this molecule looks way simpler to make it, isn't it? Now there is one more molecule. It is a levofrenol. So this is molecules also showing almost equal or the better analgesic property like morphine, and these two molecules are very simple and very easy to make it. But one thing still remaining, the addiction issues. Still these molecules are having addiction issues. Okay, so now going a little bit more further, so what are the things we need to consider? Now if you have been asked to make a molecule, make a drug which is having analgesic properties, what do you need to know? What do you need to do? So you need to design a molecule which is having a oxygen, which is a hydrogen bond donor or hydrogen bond acceptor, and you need to have a hydrophobic component or aromatic ring here, and then you need to have a hydrogen bond acceptor or the ionic bond hydrogen bond donor or the ionic interaction center. Then once you have this one, and more important thing is to have this pharmacophore triangle. When you're talking about this pharmacophore triangle, that means all these three components are important, but their positions are also very important. Just having these three components in a molecule may not be a effective analgesic drug like candidate. However, the possibility is high if you maintain the distance and the angles. If you have a molecule, you don't need to be having aromatic, you don't need to have the oxygen. Any component which is having both hydrogen bond acceptor and donor properties, van der Waals or hydrophobic properties, ionic interaction properties or hydrogen bond donor properties, then most likely you and maintaining these bond angles and maintaining these distances, then most likely the molecule what you are going to make it will show analgesic property. It may not be a drug because, as I told you, just showing a potency is not good enough. We need to think a little bit more ahead of that because, as I said, there are so many components in the drug development process. Okay, so now we learned those are the techniques what we can use to reduce the process of the drug development and to achieve our goal. Now the question is, do we have any other way, or this is it? Do we need to think about, do we need to consider any other solution, other way to achieve our target in a quicker manner? Probably yes. What are that? Have you heard about the term bioisosteres? What are the bioisosteres? Before talking about the bioisosteres, let's know what is isoster first. So isoster is a compounds or groups of atoms having the same number of atoms and electrons. What does that mean? Here are some examples: nitrogen and carbon monoxide, or NH3 minus or NCO minus. They are called isosters. Okay, I'm coming a little bit more in details about that, but before that, what other things we need to know about the isosters? There is just two laws behind this. We need to think about it. One is the hydride displacement law. What is the hydride displacement law? That means in the hydride displacement law, that means which is a Grimm's hydride displacement law, because this is the scientist who came up with this. What that does mean? The carbon, nitrogen, oxygen, fluorine, and neon is in the red is talking about if the carbon, what is the valency of the carbon? 4. Okay, what is the valency of nitrogen? 5, right? So to make it 5, so we can have carbon, which is 4, and we can have one more hydrogen is to make it 5. That means if you have a molecule containing nitrogen, if you replace nitrogen with CH group, then your molecule is going to have a similar types of properties. This is the goal. This is the things how we can change the molecule to increase the potency of the molecule or to decrease the associated toxicity. For example, if you have an oxygen and if you want to replace that oxygen, if you replace that oxygen, then you can see you can replace with NH. NH means the nitrogen is having valency 5 and hydrogen is 1, 6, which is equal to oxygen 6. Or you can replace the oxygen with CH2, which is a carbon 4, hydrogen 2, 6. So this is what called the hydride displacement law. What is another one is Hinsberg ring equivalence, because we are talking about atoms and groups. What about the rings? In the Hinsberg ring equivalence, where the benzene ring can be replaced with a thiophene or a pyridine, then also we will say it is going to be equally equivalent to the benzene ring in terms of the physiochemical properties. So when anything changes we are doing to increase the potency of a molecule or to decrease the toxicity, those groups or atoms are called the bioisoters. Okay, so now what can we achieve in bioisoters? What parameters get affected? The parameters affected by isosteric replacement is the size, conformation, or hydrogen bond formation capability, pKa, stability, hydrophobicity. So many parameters could be affected when you do the bioisosteric replacement. So why we need to do this? We need to do this because to increase the selectivity or to decrease the toxicity, improve the pharmacokinetics properties, increase stability, or to simplify the synthesis, as I discussed one of the example, the morphine thing, and to have a molecule which could be patented so that you will have some exclusive rights. But more important things in this table, you can see there are classical bioisoster replacement, which we just now discussed, and there are some non-classical bioisosteric replacements. So in the drug development process, when you are going to modify the molecule, what generally people do, oh, okay, we have this molecule in our lab, we have another chemicals, let's do the another modification and do some variations. That means there is no logic behind that. But to make it more logic based, to make it more argument based, then if you do the changes or modification based on the bioisoters, that makes a lot of sense. So here is one example. So uracil, you all know what is the uracil, right? So from using this bioisosteric approach, the fluorouracil came to the picture. This is the classical way of using the bioisosteric replacement, where this hydrogen is just replaced with fluorine. And if you go back, if you see here, this monovalent, where this any monovalent atoms can be replaced with fluorine, and this hydrogen is got replaced with fluorine, and we got this fluorouracil, which is an anti-cancer drug currently people are using. And now you can think about just a simple change, the activity of the molecules got drastically changed, selectivity changed, potency changed, and now you can think about a little bit how the bioisosteric replacements helps in the drug development process in a quicker way. When we have these biomolecules, do some changes, and then you can achieve your thing. So this is one more example, ticonazole, which is an antifungal drug. It is a topical antifungal drug. That means for the external use, if you have a skin infection because of the fungal infections, then this is the drug which is used for this treatment. Currently, this is available in the market. Now, when someone is having a systematic infection, that means someone is having urinary tract infection or some other fungal infection inside the body, is this drug can be used? Unfortunately, no, because it has a poor solubility. So now if you're going to develop a new drug completely for this systematic fungal infection, it will take ages. But by using this bioisosteric replacement approach, probably we will be a little bit easier, quicker to deal with this. How we can do that? Now you can see here what they did is they just replaced this thiophene ring with this triazole ring. So this is a non-classical bioisosteric replacement. So what they got, the polarity of this molecule got improved. So that means this can go to the systematic things. But unfortunately, this molecule is having high toxicity. So they did a little bit, the researchers did, the scientists did a little bit extra on this. And then what they did, they come up with replacing this chlorine with fluorine. In addition to that, what else they did is they replaced they keep this triazole ring intact. They just only replace this chlorine to the fluorine, and now this molecule is currently used as a drug for the fungal infections. Now you can see how this bioisotopic replacement can help us to achieve to get a drug candidate in a quicker way. So this is the one more strategy. The last one is the drug repurposing. In the drug repurposing approach, where as you have already seen in this pandemic situation, this drug repurposing approach was extensively used. That means since we do not have any drug for this COVID-19 or the viral infections, so all the possible antiviral drugs, all the possible antibiotics and all, they were tried for these infections. So for example, chloroquine, hydroxychloroquines, and so many other drugs were tried for this. Some people got benefited also from this drug repurposing approach, and some drugs got not effective at all. So why we need to do this? So here, if you look at this drug development process, in the traditional drug discovery process and this drug repurposing process, if we are going to achieve, we are going to get a new drug from here, so we can completely skip half of the process of this drug development process. We can come to the market, that we can come use the drug for this COVID-19 infection in a quicker way. So here you can go ahead a little bit more. The drugs, you can see the list of the drugs which are used currently, are considered currently for the COVID-19 infections. So among them, this chloroquine, hydroxychloroquine, and favipiravir are we consider for our further development process in the drug candidate. So what we did, we considered this chloroquine and hydroxychloroquine here, and then we took this core nucleus and we go further. But as you know, this chloroquine, hydroxychloroquine were showing some effectiveness, but it's not as effective as expected. So that when we did some computational programming, then we found out that this nucleus playing important role in terms of this dealing with this protein of this COVID-19 virus. So we use this concept and we keep this scaffold, and we try to introduce this CFT group in this ring system. In addition to that, we also introduce this triazole ring, and then we come up with these molecules. Then you can ask why I did these things, what is the purpose behind that? So here you can see this introducing of CF3 and introducing this triazole ring, and this using this another phenyl ring here. It is a bioisosteric approach. So considering this approach, we started synthesizing these molecules, started from this substituted anilines and with the trifluoromethyl acetoacetate, and then we got this ring system as I discussed, and this is called the Cornard-Limpach synthesis. By following this approach, we make this molecule, and then we use this propyl bromide and all, and then we got this alkyne component. And then in other side, we got this azide by using this substituted anilines, and then we introduced this triazole. And as you know, making this triazole, we use a well-known chemistry, is called the click chemistry. By using this click chemistry, we came up with a set of these molecules. Once we got all this set of these molecules, and as I said that we did the computational programming, in this computational programming, we identified this hydroxychloroquine and chloroquines are showing some kind of interaction to that protein. And here we use 6LU7, which is one of the lead protein in the coronavirus. And then among them, the 10Z in this molecule, the 10Z was showing more potency in terms of the computational approach. So then we did the screening against this virus. In the screening process, as you can see, all these three molecules, comparing this chloroquine and hydroxychloroquine, and you can see the 10Z, which we just now I showed you from the molecular docking approach. Here, the IC50 value is 0.060 millimolar compared to this chloroquine and hydroxychloroquine is way better. Now, the more important thing is the SI. So what is the SI means? Just the selective index. When you're talking about the selective index, because Nowadays, in the drug development process, selective index or the cytotoxicity study is one of the important component in the drug development process. What does that mean? That means when you're making a molecule and we are claiming that our molecule is having effectiveness against this virus, bacteria, or the cancer cell line, but the more important thing is my molecule is having any effective towards the normal cell. If my molecule is showing some effective or some adverse thing or killing the normal cell, then I cannot say that my molecule is a drug candidate. So that is one of the important thing at this point in the drug development process. Long time back, if you look at any articles, research articles, people were only claiming that these molecules are good, showing anti-cancer properties against these cell lines. But no one was talking about how these molecules are showing against the normal cell lines. But now at this point, this is one of the important component in any articles you will see, because that's the important factors we need to understand. For example, if you're talking about anti-cancer properties or antibacterial properties, if you take the phenyl also, what we use for the cleaning our toilet and all, and if you kill on the bacteria, they will die. Can we drink that? We cannot drink that, right? Because that will kill us too, because that will kill the normal cell. So we cannot claim that, okay, phenyl is having antibacterial properties or antiviral properties. We cannot do that. So for that, we need to understand the cytotoxicity studies. So during this process, selective index, which is talking about the potency, that means how this IC50 value and the cytotoxicity. So in other words, higher is the selective index, better is the molecule. So if you now comparing the selective index of this hydroxychloroquine and chloroquine, the synthesized molecule is having higher selective index. That gives a sense that this molecule is way better than the chloroquine and the hydroxychloroquine. Okay, so continuing this process, as I did mention to you, not only hydroxychloroquine and chloroquine we consider the favipiravir for our drug development process. So as we got a better outcome by introducing a triazole ring, then we used here the same approach by using the propyl bromide, and we introduced this alkyne part here, and then we use this azide synthesizing from the amine, and we ended up with introducing the triazole ring here in this molecule. But to diversify the pool of these compounds, we also use a little bit more approach to make more molecules. For that, we activated this carboxyl group of this piperazinic acid and with benzotriazole. And then once we activated this carboxylic acid, then we coupled with amino acids. And then we ended up with pyrazinic acid amino acid conjugates. And then this carboxyl group again activated with benzotriazole to come up with a molecule of benzothiazole and piperazine with the amino acid linker. So here, this is the molecule targeted, but we also synthesized, just skipping this step, and got directly by using this microwave technique. But in this approach, the molecules are a little bit having impurities, which we need to recrystallize and purify, and the yields are a bit low. However, in this approach, just activating this carboxyl group by using benzotriazole, even though it is one step extra, but this process way was way better in our synthetic approach perspective. Again, all these molecules, we synthesized introducing this triazole ring and introducing this benzothiazole ring. We came up with a list of the molecules, and we found out that this SI of one of the molecule, which is the 5E with the triazole ring and 12I with the benzothiazole ring, shows better SI value and better IC50 value compared to the favipiravir, which is a drug which is approved, especially in Japan, as a drug for the coronavirus infections. So compared to this molecule, these two molecules shows better activity and better SI value. So and then we consider little bit further, so since in another project was going on in our laboratory for the anti-cancer properties, but now the one thing important is I did mention to you before about the QSAR model. So since we got a good number of molecules, that means with a quinolone ring system, with the piperazine ring system and all, we developed a QSAR model out of those molecules. Once we develop this QSAR model, and there is another project was going on in our laboratory for the anti-cancer properties. We thought, okay, can we use those anti-cancer property molecules for this coronavirus things? And we use this QSAR model and try to see if the molecules are showing any kind of activity or not. So here you can see these are the molecules where we are in the process of making for the anti-cancer properties. And you can see this sunitinib, it is anti-cancer drug, and we try to make analog of this. And these three series of molecules we were synthesizing in our laboratory. And we use these molecules in the study QSR approach. Among them, these two set of molecules were showing a kind of a interesting and promising outcome, promising predicted property from this QSR model. And from keeping in that mind, we screened these molecules against the cancer cell line, but also for the against the coronavirus. Because one of the important things we need to consider at this point, people are looking for who are having cancers. If that cancer patient is getting infected with coronavirus, their life is way miserable than a normal individual is getting coronavirus because of the treatment, because they are going through some chemotherapies and they are not ready to take any kind of a drug specifically for the coronavirus. And because of that, their problem is so severe than the other common people. So people are started looking for a anti-cancer drug, which is also showing some antiviral properties, so that they can use those drugs as a chemotherapy to control these viral infections. So keeping that thing in our mind, we were trying to explore our molecules which we are trying to synthesize for anti-cancer properties. Let's see how these molecules are showing for the antiviral properties against the coronavirus. So for that, we prepared these molecules by following this synthetic route. I'm not going in details in synthetic route. This work is also published recently in the last month. You'll see that reference, and you can go a little bit more in details. So this series of molecules were synthesized by following this synthetic route, and then we did this study, the cytotoxicity studies and this IC50 values and all. And interestingly, as I did mention to you, the 6 series and the 10 series, if I go back and show you the 6 and 10 series, which we found from the QSAR model, where these 2 series are showing some promising predictor properties against this coronavirus, we also interestingly got these 2 molecules are better SI value, better IC50 value compared to the the standard drug hydroxychloroquine, chloroquine, and favipiravir. And this one recently published in the Bioorganic Chemistry. You can see the details there. Okay, so now in conclusion, so we have synthesized diverse set of heterocyclic compound using rational drug design approaches in good deal by using optimized facile reaction conditions. So as you see, we have used this bioisosteric approach, we have used this molecular docking approach, we have used this 2D QSAR approach to get these molecules to achieve our to develop the drug-like molecule for this coronavirus. So next one is some of the synthesized compound, as you see, having a better promising antiviral properties but more important thing is this, some of them are having better selective index. And as I did mention to you, the selective index is more crucial than just saying this having better antiviral property or better IC50 values. Selective index is more important because it considers the cytotoxicity properties of these molecules. Now, these preliminary antiviral properties is encouraging, as you see. And the more important thing here is now we have the QSAR model, which could be used further to develop a new molecules. Currently, my lab is working on synthesizing a new set of molecules, keeping all these parameters in consideration, and hopefully in a month or so we will come up with a better set of molecules which are better than what we have recently published. The initial investigation data and the computational studies we are using, but anyone who want to use in the drug development process, they are free to use. They are now published in the three different journals recently, all the three works, as you can see from the presentations. And I believe those tools could be a good, useful for the further drug development process. And at the end, I would like to thank my university, Augusta University, and my students who did a lot of work on to achieve these things. And thank you, Lata, for inviting me to share some of my work. And thank you, everyone, the organizing committee. And I'll be more than happy to answer any of your questions.
Thank you so much, Dr. Shiva. It has been indeed very wonderful to have such a knowledgeable talk from you. As we all know that this pandemic has really shaken the world terribly. And sir, your talk has been indeed the need of the hour. And as I mean, it is evident from the number of questions which have been flooded in the chat box. I can see that how our students and the participants, they are so much enthusiastically and they have been involved in your lecture. Since you have already summarized your talk, so I would like to thank you for this stepwise. You have introduced the concept of drug designing, starting from morphine, and how the latest technologies, like you have talked of QSR and molecular modeling, they have, in fact, as we say in chemistry, they are working as a catalyst to uh, expediting the process of drug designing, right? So the introduction of bioisosteric replacement, all these things, I think they are very useful. And you have you have explained them in such a simplified way.
Ma'am, you are on mute.
Sorry, just I'm so sorry. This has been really good at this time to have such a knowledgeable talk. So I would like to take some questions which are there in the chat box. One is that is molecular hybridization is always good for screening the molecules for searching the drug molecules? This is a question by Rishika Arya.
Well, the molecular hybridization is always better because most of the times the molecular hybridization approach means two different molecules which are having two different targets, and we assume that this molecular hybridization can work as a synergistic effect. But the more important thing here is molecular hybridization could be acted so differently. So that means if you are having two molecules, how you are linking these two molecules. For example, if you are linking these two molecules by, let's say, through the ester linkage. So if you have the ester linkage approach, then we can say that is not going to have a synergetic effect, because that's going to act as a prodrug. Because when you are going to take this molecule, as soon as it will go inside the body, because we have a lot of esterase enzyme in our body, immediately that ester bond is going to break. Now you will have these two different molecules going to act differently, separately. It's just like two different pills you are taking for that purpose. But if you are using a different type of linker, which are not going to be easily breakable because of the common enzymes which are present in our body, probably in that way we can see differently. But more important things why I just wanted to let you know here is, if your approach is kind of a pro-drug, then in that case you may not see a good outcome in the in vitro studies, but you will see better outcome in vivo studies. But that is the two different things. People are getting confused, people are getting discouraged when they do in vitro studies. The activities, no, they are not there. However, in vivo studies, when they, because of the discouragement, they do not go for in vivo. But most of the time, if you have that approach, when you go to the in vivo studies, you will see better activity. I hope I answered your question.
Sure, sir. So there's one more question. What is the most important or crucial part of drug development process?
Most important crucial part of the drug development process, I believe, is to identify the lead compound. Because once you have that lead compound, it is way easier to modify it, way easier. For example, I'll give you one example. I guess we all know about the malaria, right? And when the malaria comes to in our mind, which drug we can use for malaria? Immediately in anyone, if you can ask, even though they are not know about, they do not know about the drugs and all, they will say quinine. Quinine is the best drug for the malaria. Quinine was isolated and identified 200 years back. Till now we are using. But the more important thing is whatever the drugs are developed after that. You can say chloroquine, mefloquine, or primaquine, and all are drugs which are used, which are resistant to the quinine for the treatment of malaria. All molecules are having the same structural scaffold as quinine. So the drug development for the chloroquine, mefloquine, primaquine, and whatever it is, the drug came for the malaria, for the drug development was so easy because of the structure of the quinine. So for them, the quinine was a lead molecule. If we do not have the quinine structure, think about from where we will start to make a drug for the malaria, for the treatment of cerebral malaria or any types of malaria. For that, I believe the lead compound is one of the crucial steps before for the drug development process.
So same on this, there is one more question, that how anti-malarial drugs are used for against coronavirus.
Well, there are so many other factors where it's not very much clear yet. And as I said, that chloroquine, hydroxychloroquine, were initially were considered, were claimed that these molecules could be a good properties for for the coronavirus, but later on they said that these molecules are not very much effective. So for example, this is not directly correlated the anti-malarial properties and antiviral properties, but they are having diversified biological properties. For example, I will give you one example. Probably you have heard or you have used the drugs called the ciprofloxacin and norfloxacin, which are antibiotics, right? But currently, people are using the ciprofloxacin and norfloxacin for the treatment of cancer. But recently, they identified these fluoroquinolone antibiotics are also having cancer properties. In my lab, we are working on to modifying those ciprofloxacin, norfloxacin to increase the anti-cancer properties. So sometimes we do not know, once we get these biological properties, we stick to there, but sometimes some molecules are having some other effectiveness. This is called the repurposing, drug repurposing approach. So because of this pandemic and we are having a scarcity of drug for this pandemic, we or the doctors were tried all possible drugs and they just reported whenever whatever they have observed. Initially, they found that okay, this chloroquine, hydroxychloroquine, when they used, they found the patients are getting a little bit better. So there is no rational and nothing exact or physiology or exact pathways are known yet. There may be something, but at this point, there is nothing clear yet.
So there's a question that why we are vaccinated twice if they have similar functions?
Well, the vaccination twice, first of all, why we are vaccinating? We are vaccinating because we need to have the antibodies to deal with. the when we are getting infected with this virus. The thing is, the antibodies what we have present in our body, that is going slowly, the amount of the antibodies going down. That means that your immunity towards this coronavirus is going down. So for that, we need to have a double dose. Now, last week I took my third dose of the Pfizer, which is called the booster dose, because they says that after 6 to 8 months, the antibody content of is going down. So you need to have third one to maintain that antibodies. For example, if you are, I'll say this one, you're taking ibuprofen for pain. Why you are taking ibuprofen every 4 hours? Exactly, if you understand the concept of that, exactly this. But some vaccines you do not need in a lifetime. For example, measles. If you get a vaccine for measles, or in your lifetime you have measles, then you do never need a vaccine for that, because your body is keep on producing that antibodies for that against the measles. So every antibody is different, how your body is reacting. But at this point, the antibodies what we are getting from this vaccines for the coronavirus, our body is not producing. We need to have depend on some external sources, and those are amounts are slowly getting reduced in long time. So that's the one of the reason we are going to take second dose, probably third dose.
I hope Gaurav, your question has been answered. Now, there is one thing, that what is the effect of functional groups on activity of any drug? Somebody has
So
asked.
that's actually really a good question in the drug development process. Functional drugs are kind of a if I just tell you in kind of in the layman language, so you can say if the two friends are going along, what is the importance of holding their hands together? That's the exactly things what is the role of the functional group. The functional groups are interacting with the receptors, the protein. When you take the drug, the functional groups which is going to react or going to interact with the receptor and hold there, because if your drug is not going to stay or hold for the longer time with the protein or the receptor, you will not see the effect. If it is just a bypass, then you can say that if the two persons are not holding their hand together, then you cannot say that they are best friends, right? You can just say hi and pass away. Then you can say that, oh, they are just friends. If they're holding, then you can for sure say that, oh, they looks like they are best friend. Exactly the same thing happens when the drug you can take, then the drug is interacting with the receptor. If the drug is stay there for longer time, then you will see the effectiveness, because it is going to inhibit that or kill that virus and whatever it is. Then that kind of interaction is needed because of the functional groups. So functional groups are depends on the need, as I discussed before in the molecular docking approach. So when your receptor is having a hydrogen bond donor in the active site, you need to design a molecule with a functional group which is going to have a hydrogen bond acceptor. Then a donor acceptor will interact and stay there. Or a ionic property, then you need to have something in your molecule in a functional group which has that ability to do the ionic interactions. So this is the importance of the functional group. That doesn't mean that any functional group will give the biological properties. It has to be complementing to the receptor.
And so one last question, which is there, that what is applicability domain in QSAR studies?
Oh, I didn't get the question, please.
It says that what is applicability domain in QSAR studies?
Okay, so the applicability domain means in the QSR studies, we need to understand one thing. In QSR studies, that is considering only the structural parameter. It is nothing related to the receptor or protein. It is completely related to the uh, structural components. So this is one of the important tool. When you have a QSR model, you can design any molecule and you can use in that model. If your model is going to match the new molecules property and with the database, then the model is going to come up with a prediction that your proposed molecule is going to have a better activity or not. If the program is going to say, no, your molecule is not a good fit for this specific type of issues or specific type of disease, then probably you need to modify the molecule or something. You can do some changes and then again re-upload it. So this is kind of a tool which will reduce your synthetic effort. You do not need to synthesize a molecule until you get a kind of a green signal from this tool. There are so many free tools are also available if you Google it for the QSR properties.
Thank you so much, sir. I think this participants, they have got the answers to their questions, and it was a pleasure to have you with us on this platform. Thank you once again from all of us for such a wonderful talk. Thank you, Dr.
Thank you so much for your time, and thank you so much for inviting me. I hope you will have all a good day.
Thank you, sir, for sharing your knowledge with us on different strategies used for searching the potential molecules against the coronavirus. And I'm sure that our students get motivated and enriched with the concept of heterocyclic molecules and their use for the development of novel drug candidates. Now, I request Dr. Pratibha Chaudhary to welcome and felicitate our invited speaker, Dr. Upendra Kumar Singh. Now, over to Dr. Pratibha.
Thank you, Lata. Good morning, everyone. Myself, Dr. Pratibha Chaudhary from Department of Chemistry, Maitri College, welcomes you all to the second day of the international conference. It is my great pleasure to introduce the speaker of the session, Dr. Upendra Singh, Senior Immunologist and Postdoctoral Fellow at Hogan Lab, Center for Autoimmunity and Inflammation, Lajola Institute for Immunology, USA. Today, Dr. Singh is going to deliver his lecture on topic CRAC channel importance in the regulation of cell function and during channelopathies. He completed his BSc and MSc integrated in biotech and bioinformatics from Center for Interdisciplinary Research in Basic Sciences, Jamia Millia University, Delhi. In year 2018, he received his PhD in biophysical chemistry from CIRBS, Jamia University itself. He has established a wide interdisciplinary collaboration network and has published 15 high-impact research papers in various prestigious journals. He has more than 11 years of work experience in studying the several different aspects of store-operated calcium channels, such as the mechanisms that regulate their activation and inactivation, regulation by oxidative stress, modulation by chemical compounds, and various others. He has a vast research experience in protein biochemistry, biophysics, biophysical chemistry, chemical biology, cellular biophysics, and molecular biology. He is a life member of Indian Biophysical Society and recipient of awards in his professional career. He is professionally expert in various lab techniques also, like DSC, ITC, protein purification techniques. He has also worked to characterize the mechanism of TASA aggregation in bacterial biofilms and role of inhibitors in the Institute of Chemistry, the Hebrew University of Jerusalem. Currently, he is working on store-operated calcium entry mediated by STIM and ORI proteins. Dr. Singh, an expert in the field of drug design and biophysics, is present among us today, and I'm sure that we are going to get enriched by his knowledge and experiences. On behalf of all the participants here, I welcome you, Dr. Upendra, once again to this international conference. Thanks, and over to you, Dr. Upendra.
So, thank you, Dr. Pratibha Chaudhary. I'm very for this nice introduction. And basically, I'm a biophysicist, which I have done my PhD and move on for biophysical chemistry at Hebrew University, Jerusalem, and then few years back, I have been working on the crack channels at La Jolla Institute for Immunology. It's a immunological institute in San Diego, California. So yeah, good morning, everybody. And I'm Upendra. I'm going to talk about the crack channel importance in the regulation of cell functions during channel pathways. Okay. So first I will start with the calcium signaling. What is calcium signaling and why the calcium signaling is important? So being one of the second speakers who is talking about the calcium signaling, I know that you people already have known a little bit of basics about it. So calcium signaling is a ubiquitous process. It occurs in many and all type of cells in our body, whether it be neurons, whether it be skin cells, whether it be retinal cells or the liver cells, cardiac cells, all muscle cells or the smooth muscle cells, arteries. Calcium signaling is a very important part in every part of our cells in the body. And there are many types of calcium channels which are present throughout these plasma membrane, ER, and various others. So namely, what are the most important the calcium channels are the voltage-gated calcium channels. Those are mostly work on the basis of the voltage depolarization, and the voltage gated works on the basis of voltage applied. So these channels are mostly, you will see that they can be measured electrophysiologically, and they're very important because they are present in the heart muscle cells or the muscle fiber cells. The extracellular conditions outside a cell for the calcium is around 2000 micromolar, that is 2 millimolar. And inside in the ER, it's like almost 600 to a millimolar concentrations. But in the cytoplasm, it's much devoid of the calcium concentration. If you see, there is very less calcium. There are many reasons for it. And one of the main reasons is that calcium concentration can trigger the cell functions towards death or life. So it should be tightly regulated for various processes in our body, important biological process, I mean. So some of the channels in the plasma membrane and the ER I have shown on with this image, in which it shows that the calcium lower concentration are also absorbed by the sponge proteins. Those are calmodulin. It's very important proteins. So today I'm going to talk about the crack channels, which is the comprised of the ori in the plasma membrane and stem in the ER. And one of the important addition to this crack channel, which is not a part of the crack channel, is IP3R, which maintains the process. So let's start with that. So what are the crack channels and what are they called? The crack channels are the calcium release activated calcium channels. These are comprised of the ori in the plasma membrane and stem in the ER. So the crack channels are well characterized because they are very much important and one of the only store-operated calcium channels. Store-operated calcium channels are the channels which work on the basis of the calcium concentrations, as ER is the calcium storehouse of the cell. So whatever the calcium have to be pumped inside the different organelles or in the cytoplasm, ER serves as a very important store. So stem is a very calcium sensing protein in the ER and is responsible for the store activation. So whenever in the resting T cells, we can see that the stem here is kind of situated in the ER far from the plasma membrane and the ori. And when these calcium channels are activated, the stem is the crack channel activated, the stem goes back and bounces to the ori and forms a channel. So basically, yesterday, Rajesh have introduced a lot about the a little about the different types of store depolarization and activation with the calcium signaling, and a little bit of about the store operated calcium entry too. So I will mostly focus on how the stem behaves and crack channels occurs. So this is a STIM1 activation I'm going to show you over there. This comprises of the EF hand in which the calcium binds, and it is the EF hand is the site the E luminal part of the STIM1, which is located inside the ER lumen. This is a transmembrane helix, in which is the most important, which comes together upon activation, and thereafter is followed the CC1 coil-coin-1 domains and the SORCAT domains, which are folded back when there is no store depletion. So what happens where store depletion occurs? Store depletion is a process in which the calcium is devoid of the ER lumen is devoid of calcium. It decreases much below 600 micromolar, around like 30 to 40. What happens? Calcium detaches with this EF hand down there, and they go back, and the CC1 complexes come together, form disulfide bond, they dimerize, and it extends to the membranes of the plasma membrane. It tightly packs with its with the C terminal, which is the polylysine or poly K terminal we say about here. It forms the crack channel with ori, which we can see over here. So this is the total process, how it occurs. And then the calcium directly couples and enters into the ER lumen, and the store gets repleted again. So this is the overall process, how the store operated calcium entry works. So this is the image I'm going to show over here. In this image, we can see the GFP labeled STIM1. The GFP is labeled, the green fluorescent protein is labeled in the amino terminal of STIM1 and is transfected into the HeLa cells. And these cells having the GFP can fluoresce. Because this GFP have a property when excited 488 nanometers, they fluoresce as 510 nanometers. So excitation was collected at 510 nanometers in the live cell imaging. And when the stores were this is the resting stage of the STIM1, in which the STIM1 is folded back onto the ER, and they do not dimerize specifically. So when they are added with thapsigargin, now thapsigargin is a irreversible inhibitor of the SERCA. So once added, it reduces the SERCA to pump back calcium into the ER. And after a while, what happens, the calcium leaches out outside of the ER, and due to detachment of the calcium from the EF hand of the STIM1, they come together, dimerize, and form puncta. So we can see clearly over here that the puncta in this small dots over here is the activation state of the STIM1. So the first step in STIM1 activation is always the dimerization. So it dimerizes, helps get extended, holds the crack channel, holds the ori channel, activates the ori channel, and after formation of crack channel, the calcium is repleted into the ER stores with hundreds of thousands of calcium ions per seconds, and then the store gets repleted, and then they're folded back and again in the resting stage. So this image is a very nice visualization of the STIM1 domains, how the STIM1 is distributed wide enough into the ER, and then get clustered as a forming junctions of the ER plasma junctions around with the puncta. This is the calcium signaling, the calcium imaging with the calcium signaling measured with the dyes at the different ratios, in which we see when we start with a cell which have no calciums around it. So we choke or we add a chelator, calcium chelator around the cells when we visualize with the microscopy, in which we add the zero calcium buffers, or having the zero calcium ringer buffer, having the EGTA, which chelates calcium and completely maintains the extracellular condition of calcium is to be zero. Then when we add the thapsigargin to these cells, these irreversibly chelates the irreversibly binds to the circa and depletes the ER of the cell. And when we use or replete the extracellular conditions with Ringer buffer having 2 millimolar calcium, it increases in intensity, and we can see that the cytoplasm increases in number of calcium molecules, ions over here. And the one which do not have, they do not show any STIM1 activation. So one of the simple examples which have been published from our lab, that this is the behavior in which, this is a control experiment in which the EGFP labeled STIM1 when activated goes to the channel and the STIM1 goes to the junctions and shows formation of puncta. And there was a mutational construct 2NQ. This 2NQ construct is a mutational construct of STIM1 in which when we add or when we replace the charged residues to the neutral residues, 10 charged residues come from 160 amino acids to have 210. It is the major binding site of the calcium into the ER lumen other than the EF hand. It did not, the stim turn goes back to junctions. It's kind of now unaffected by the adding tapsigargin. It's not getting activated because the calcium binding sites have been mutated. So this experiment shows how the STIM1 binds to the calcium, and a nice visual explanation of how the STIM1 gets activated and goes to the junctions and conduct the calcium signal. So this now, why the crack channel? We understand the crack channel now. Now we have to understand why the crack channel is important. To show the importance, I have put on some of the slides which are from the which shows the importance of the immune tolerance in the functions in which the store-operated calcium entry in T cells here is very crucial for generation of agonistic selected regulatory cells in mice during the development of the thymus. So when store-operated calcium entry occurs, the T regulatory cells gets activated and divide into follicular or the tissue-resident T regulatory cells. Now, the inactivation of SOCE in thymic T regulatory cells after the thymic development shows an additional role of the crack channels in mature T regulatory cells. So we can tell that the SOCE is very much important in the immune tolerance of the T cells in formation of T regulatory cells. For the people who do not know the T regulatory cells, these are the cells which are kind of check the overactivation or the reduces the immune response. So now the other functions of in which the apoptosis, the importance of apoptosis in with the SOCE, which shows whenever there is like the calcium entry from the crack channels, it can activate or production of inflammatory cytokines such as the interleukins, IL-7, So this one is the calcium influx, which triggers the induced cell death, AICD, and induced by facial trail and expression addition of store-operated calcium entry, which regulates the intrinsic apoptosis mechanism by expression of the BCL2 families. So the calcium which binds to NFAT produces BAC and NOXA enzymes, which helps in the apoptosis and directly killing off to the cells. So when here is the autoimmunity and inflammation, which shows that the store-operated calcium entry, which with increase of intensity of store-operated calcium entry, are very much important for the granulocytes produced like IL-17A, IL-17F, GM, CSF, and with these cells, the T regulatory cells, which shows the suppresses the immune response, and this one which shows the therapeutic window of the distinct SOCE functions in which the cell subsets are kind of produces the autoimmunity and inflammation, and they can differentiate it into different cell types. So here, this last image shows the immunity to viral infections requires store-operated calcium entry in both CD8 positive and CD8-4 cells, in which the ORI, when enters, it kind of this transcription factors bind to this cells, which can perform very specific functions in the perforin Graham's enzyme. These are the very specific enzymes which are regulated for the release of calcium molecules again back into the cell, into the extracellular space. So these cells store operated calcium entry mechanisms are important of the development of different type of T subsets as well. So one of the last functions I'm going to talk about, the crack channels, in which it sums up all the functions, is that the store operated calcium entry, which shows that all the TCR, the calcium, the crack channel formed with the ori and stem, enters the calcium into the cells, into the ER, and the TCR gets activated by IL-2, which activates the phospholipase C, and it forms the IP3 molecules, which can bind and release the ER from the calcium from the ER into the cytoplasm. And this calcium into the cytoplasm can be used to activate the calcineurin-like proteins, which dephosphorylate NF80, and this NF80, and as well as it acts with the KMK2, NF-kT beta, and other mitochondrial functions, and here the NF80, after dephosphorylation, it binds with different interaction partners and are really important for targeting of genes. One of the nice functions is interacting with AP1 and getting targeted with IL-2 genes. And this is a function of mostly the immunity related, in which there are various other functions as well, in which it interacts with MAT3, FOXP3, and the target genes help to regulate or form different type of T cells, like T regulator cells, 1, 2, or 17 T helper cells. These are T helper cells are kind of CD4 positive cells. So why channelopathy so this is the importance of the crack channels, which regulates completely the transcription over into the cells. So important question is, what happens if we encounter a channelopathy or the loss of function of the stem or the ori? So the most important thing is that the crack channelopathy, the stem ori functions is abolished. And it is a disease, or it is a condition which is autosomal recessive. We can call this autosomal recessive because whenever there is, it's mostly shown through phenotypic contributions of the cell. Whenever you have to see physical proof of it, like like in SCID patients, like not development of any specific subsets, something like that. So autosomal recessive hint towards the behavior in which both of the genes should be dominant to express a phenotype. And if they are like not dominant and recessive, they definitely will not express a phenotype and will be heterozygous and will be carried on to the generations and will be moved on in the offsprings. So crack channelopathy is kind of defined in the combined immunodeficiency, in which there is a severe any pathogen infections. And they start in the earliest part of the life. It's not like they have to start in the very late part of the life. They start very early if they're very related to the immunodeficiency. And that happens with loss of ori and stem. Now, they have also been associated and defined with the autoimmune hemolytic anemia, muscular hypotonia. This muscular hypotonia is the myopathy in which what happens that the muscle do not grow properly, or the muscle grow very irregularly into the body, and they can be seen. It's a phenotypic thing. It can be seen pretty much clearly. It's mostly associated with the STIM1. The other part is the ectodermal dysplasia, in which the dentate enamel classification or the sweat gland function do not happen. So the last three are not very life-threatening, but the combined immunodeficiency is a kind of life-threatening over here. And this can be cured. This can be done with a kind of transplantation, which is the hematopoietic stem cell transplantation. But that if you take a wild type cell and put it into the wild type stem ori, into the of normal individual, which having the loss of functions like ori, they develop and they produce nice calcium response. So they are not only loss of functions, they are gain of functions too, and which the autosomal dominant gain of functions mutations occur in the ori and stem and causes the tubular aggregate myopathy. This is also a skin muscle fiber disease in which the muscle fibers are like type 1 and type 2. The type 1 are the slow ones and the type 2 are the fast ones. The type 2 are the fast twitch, which produces the fast response. So whenever you lose the type 2 response or the type 2 fibers, you develop only type 1, and there will be a problem in which has histopathological evidences, which having the clots or the clumps or by biopsy that can be seen as a form of aggregates. That's a pretty difficult kind of thing. So combined CID is ORI in the ORI1 and SIM1 mutant patients. They have the normal number of immune cells in the body, with exceptions of reduced regulatory T cells, which reduces immune response, and natural killer cells, which are the part of the adaptive immune response. So combined immunodeficiency is a kind of serious conditions. And the ORI stem mutations result in imbalance of immune response with the concurrent defects in affected T cells. This all we can tell about from the previous slides, which how the calcium channels, the crack channels are important, or the store operated calcium entry is important for the calcium into the ER, and as well as all the most of the regulation functions in the related to immune cells. So I would like to talk about one of the conditions, which was the crack channelopathy, which was first mentioned in the Stephen Fiske paper, in which there is a condition. They tried to, there was a family, they draw a pedigree over here. The pedigree, which having the diagonal crossed, clearly shows that they are, the parents are deceased. So the authors were not able to monitor the kind of, the immune or the heterozygous or homozygous conditions where the carriers are not. The round ones are the females, and the squares ones are the males, and the clear ones completely show that they don't have any recessive or dominant. They're not the carriers or the expressing any disease. And the black ones completely shows that the disease, they are diseased one or the SCID ones. This with the half black and half white circles, this shows that these are autosomal recessive. These are the kind of heterozygous that carry the disease and spread it into the families or carry on to the families. So the pedigree of SCID patients with the defect in the store-operated calcium entry is shown over here. And the samples 8 and 11 shows that these were the SCIDs, and the the half-shaded ones are the heterozygous and the carriers. So this pedigree was done by taking the samples of the 4 generations people, the people from the 4 generations almost, like it was the study conducted in Harvard, and Stephen was the prime author on this paper. And it clearly shows that the marriage between the far family cousins developed the SCID, and it was a kind of mutations related in the exon 1 of human ORI1, in which the nucleotide changes from C to T and the protein changes from arginine to tryptophan with one single nucleotide, single polymorphism. And what happens that the amino acid which it's code for changes to tryptophan, and this disease and the whole ORI1 channel gets inactivated, and due to which the disease is carried within families, and then it can show a very difficult condition, or it develops into disease with very low immune response and eventually the death. So the authors after mapping this polymorphism, they took the cells out of this body and tried with every 21 family members. And they found that 13 of the 21 family members they were heterozygous, and they showed a different response where in the 0.5 millimolar calcium, the response, the spike in the after adding calcium, the increase in that the calcium response was reduced. But it was not observed previously in 2 millimolar calcium, but it was observed in 0.5 millimolar calcium. This shows that the 13 members of the family did not have a proper function of the crack channel. So this is something which was the first reported study of the crack channelopathy. So over the years, it has been seen in the human, the ORI and STIM expressions of the stored operated calcium entry is quite different. This is the wild type in which the calcium imaging experiments completely shows the wild type cells produces in 2 millimolar calcium re-addition, replace the cells and the cells and replace the cells with the calcium. But in the STIM1 and ORI loss of mutations, there was none in humans. So STIM1 is only associated with one of the disorders. The major one is the ORI one, which is to be cared for. So STIM1 has, as from the previous slide, we can see there is only one mutation. This is the E130X. So in the mouse, compared to the human model, in various mouse models were developed having the STIM2, STIM1, they're the floxed gene in which the STIM2 and STIM1 were kind of genetically engineered and removed from the expression level of those animals. They were not expressed so forth. So they have a different level of expressions. The wild type was ORI2 was expressing more than this wild type, and STIM1 and have a lower expression compared to the other ones. So this clearly shows that the STIM1 is quite important in the signaling molecules, and the human and mouse STIM1 or the crack channel deficiency responded in a very different way. But this is what is shown from the T cell functions rather than shown from any of the other cells. Other cells might behave differently. So now we understood that there is a crack channelopathy and there is an immune response deficient with the, accomsted with all those channels. But we know that the loss of functions from the SCID patients can be treated with transfecting or the transplanting the hematopoietic stem cells with the wild type cells, and they respond properly. But what happens with the heterozygous cannot be seen so clearly, and they responded pretty much well, because if there is a phenotypic conditions in the adulthood, their enamel or the dental or other things have developed pretty much well. So it's not a life-threatening conditions. So there should be some way out for it. So for the gain of functions, in which what happens in other diseases like acute pancreatitis, the gain of functions of ORI crack channel, what happens that a lot of calcium is pumped into the cell. So how to inhibit that? That's why the gain of functions was to reduce that kind of stuff. The crack channel blockers were developed, and there are like many crack channel blockers. There are like 1, 2, 3 subtypes there. The most primary are the dihydropyridines and the others too. So this shows that the calcium channel blockers, which are mostly used for the patients in the one with the high blood pressure or the heart problems like high arrhythmia, heartbeat, or the heart pain, that is the angina, those related to it, it is mostly directly kind of related with the calcium channels in the heart, and those are not the crack channels, those are different channels. So those ones have many side effects like constipation, dizziness, fast, fatigue, flushing, and these are some of the channel blockers which I just wanted to mention. Is isradipine, nisolidipine, and verapamil. These are the green ones are from the dihydropyridine, and these are not from the same family. These are non-dihydropyridine. So the question was that if these are all the channels which these are the blockers which do not act and the crack channels, what happens if we can they act on the crack channels and reduce the gain of functions? Yes, they can. Sometimes they can. We have to mix with different crack inhibitors, and we can try. Maybe they do not bind to ORI1 or STIM1. They can bind to different channels and they can have the functions, but there is always a risk. So there is a new startup in the San Diego, and that is the Calcium Medica. This is a very great company. It's a new company just targeting the STIM ORI crack channels, in which they have kind of made many crack channel inhibitors, and most of them are as the kind of in the testing phase, in the phase 1, phase 2, phase 3 trials. So I have borrowed some of these slides so that I would be able to show some of the products they have developed and which they're still in trials. And those are like CM65, CM6325. This is the kind of Oxora. Oxora is the prime drug they have developed over the years. And those Oxora is, we will talk about Oxora later. So let's talk about CM6325. This is the electrophysiological experiment in which they have clamped the cells and they treated CM6325 to specifically check it is a crack channel inhibitor or not. This is the IC50 value for the crack current they are working on. There's like 33 nanomolars. And with addition of 1 micromolar, they show a very rapid calcium blockage, like crack blockage. They definitely block the crack current, and it shows a very potent inhibition in the native crack channels. So this result was also, this is the slide which I borrowed from the K. Startman. This is the prime data from the review. And this one, which is from the fluorescence graph, in which they show the fluorescence intensity and coupled with the treating with the CM6225 in the naive CD4 positive T cells and Th1 activated T cells, and both show very similar IC50 values. That the idea of showing this drug for the crack channel inhibitors was not just that, that blocks the crack channel. The thing is that whenever we proceed with an experiment, we know that we got an compound, a drug, which inhibits a particular pathway. What we need to do is we need to check first into the cells. And there where the preclinical or the kind of experiments, our main experiments begins. So we check the cells first with the compound to check its activity. So if it have an inhibition activity, we will move forward to the mouse models or the different animal models. And after, this is a preclinical trial. And when we get a very satisfying results, we move to the phase 1 trials. But those phase 1 trials are very much in kind of the rules for these phase 1 trials is laid down by different agencies. In US, it's FDA. So they have kind of laid down the rules for the experiments. What are the rules of the experiments, where the experiments need to stop when there is any complications in the phase 1 trials. So starting again with, there is one more drug which is synthesized by the Calcium Medica. And this was independently synthesized by Calcium Medica, and they have entered the clinical trial now. This shows that it is an acute pancreatitis conditions, and in which this is a control, and the compound TLCS and FAEE, these both compounds were added to create acute pancreatitis from the mouse in which the pancreas was extracted, and CM128 was added in the early and late stage of both the compounds and checked for. Clearly, there was a difference. There was a difference that it saves the acute pancreatitis from the acute pancreatitis. The thing is that they have also published this data in this news flash, in which shows the Calcium Medica have a positive data from the phase 2 treated in Oxora patients. So this is how the clinical trials proceed. So acute pancreatitis conditions was that whenever there is problems in the calcium channels and there is a pro-inflammatory conditions, those conditions can be or kind of impair the pancreas and also kind of hinders the lungs. And there where the SARS or the acute respiratory syndromes kind of comes into place. This is the similar conditions which occurs in the COVID-19 patients, in which the virus, when infected in the lower bottom part of the lungs, they create conditions in which there is acute respiratory disorder. So Oxora was the drug which is developed by the calcimedia. They have tried and they have been a little bit of successful with that, and they're still in the phase 2 trials. Now, the news what they have shared is, I have pasted over here, in which it shows that the interim analysis indicate that Oxora is effective in severe COVID-19 pneumonia patients. This was the news published from the European Journal, in which they have shown that with combining with the low flow oxygen therapy, the Oxora reduced the proportion of patients put on ventilators over by 50%, and that's a huge number compared to the associated medium. So Oxora was never invented or kind of found as a drug for the COVID-19, but it came to be because COVID hit at the same time. So it was first published with the clinical trial results in 2019, and where it was shown that when it's added or they were intravenously injected, they inhibited the crack channels very much and prevents the acute epithelial or endothelial cell injury and inflammation in organs, in pancreas and the lungs. And that was exactly what was happening with the COVID-19. So the people thought, the scientists at Calcimedia and the more of the kind of collaborators thought, why not give it a try for the COVID-19 patients? And it was true, and it made successful. So that is how they started with the acute pancreatitis chilling conditions for crack inhibition, but they ended into the COVID-19. Now, some of the points are that the crack channel activation in pulmonary endothelium is linked to the breakdown of alveolar capillary barrier. And the crack channel activation also initiates the production of release of pro-inflammatory cytokines from the immune cells. The resulting development of the pulmonary edema and hypomixia, and ultimately the ARDS, which is the respiratory syndrome, contributes to significant morbidity and mortality. So this is why the COVID-19 patients get to the mortality at last, or get a higher mortality rate, because development of acute respiratory syndrome, and that was the problem. So as we see that the Oxora also never originally invented or discovered as a drug which intravenously kind of in formulation binds or inhibits the crack channels. It does that with its secondary attributes. As we see that it do not have the primary binding site, or it do not compete with the virus sites or with other drugs which are kind of regulated in the dexamethasone and various others. It directly is very helpful in development of the, or the kind of treating patients compared to the standard care. So this is the progress of the drug, which have been in different diseases, and they have shown the indication and the delivery system, in which the CM6018 was only known to be the oral dosing, which is used in the preclinical toxicology and testing studies. And all the models have at least passed out, most of the models have passed out the phase 1 stage, and they are already into the phase 2 stage. And the one with the Oxora 4 for the COVID-19 pneumonia have completed very recently the phase 2 trial stage, and they have entered into the phase 3 stage. Maybe in the phase 3 trials, they can show that the Oxora can give an additional benefit, what you can get from the standard healthcare using anti-inflammatory drugs. So the point here is made that how we progress from the target of a single drug to its multiple uses, how we can move from when we get a single drug to check into the cells and to the mouse models and eventually for the preclinical studies with humans, and how it develops over a period of time, because the crack channels were first discovered in 2005 or 2006, like 1.5 decades before. After like 10 years, the mouse models were successful. We know that everything works well, and the testing began. And the company was set up in somewhat around 2007, the Calcimedica. And this is the only company which makes the crack channel inhibitors. And then they have devised right now, they're still in the the phase 1 and phase 2 trials. Hopefully in a couple of years, they can make it and we may be able to use it properly. So these are the news which have been flashed on the Calcimedica webpage, which shows the how the Oxora has rapidly reduced the inflammation and reduced damage to the vital organ tissues when used in the COVID-19 patients. And the patients which received the Oxora were better tolerated and can eat solid foods, show less severe respiratory syndromes, inflammatory respiratory syndromes, which occurs from the acute pancreatitis, and reduced hospitalizations. And all the patients received pancreatic contrast enhanced in the computed tomographs at the last of their discharge whenever, and they have seen a wide or kind of a huge improvement. So the data is like for the standard care, it was around like 75 to 80% in the patients with the mild complications, and the patients which have like very higher stage, the data was around 30 to 40% for the mild or for the moderate to high stage, in comparison to the standard care which have been offered, like putting patients onto the ventilators and staying for a while and let them the body to recover and recover themselves with the with a standard care. So I will conclude my talk now, and I will thank Professor Patrick Hogan, my supervisor, in which he have shown he is also a founder of the Calcimedica, a biotech startup company in which they are making the calcium inhibitors channels, calcium inhibitor products, and he is the one who is a person which was discovering one of the panel in the people who have discovered the crack channel, STIM-N1. So I'm very, very thankful to the organizing committee so that they have made a very difficult, and they have tried very hard to bring people onto the stage and join people around the globe on different disciplines working on different subjects. I would be very glad. I'm very glad to thank the organizing committee and everyone listening to my talk, devoting a lot of their precious time to it. Thank you.
Thank you, Dr. Rupen, for such an informative talk. And we are really, we appreciate the efforts which you people have put forward. And as I said earlier, that this pandemic has really brought a lot of change in our lives. And it has been a great challenge for all of us, and especially for researchers and scientists. And you people have been continuously working in this field. And today you have introduced to us, you started with the crack channels, you introduced what are these crack channels and what are their functions, and then we moved to the crack channelopathy and what are the different type of diseases which may result out of this, and how what are the work which are going on in the field of drug designing, which will help to come out from these shortcomings which are there. And we talked of calcium channel blockers also. So I think a balance is required everywhere, as it is evident from your lecture that you need calcium, but at the same time, excess of calcium is also going to do something. So a balance in everything, whether it is in your body or whether it is in the environment, is required. And as we have seen that I think I can say the hashtag nowadays is immunity. So everybody is very worried about what of their immunity and what is happening. And you have so beautifully explained that how this immunity is related to the very simple thing. We talk of calcium, the moment we start studying our science and everything. So that has been related so beautifully. So it is, thanks a lot for sharing your talk with us and making us aware of so many facts. And I think it will benefit our students and they will be inspired by what you people are doing. And I really appreciate the work which you have done. And it is being a real serving to the mankind, what you are doing, Dr. Rupin. Thank you so much for that. And with at the same time, there are some questions, few questions which I would like to take from the participants. And one has asked that what is transmembrane helix and what is the work of this helix? We talk.
So this is the question. This, like I was talking about the STIM1 molecules. STIM1 molecules, which are the molecules which activate the ORI channels into the ORI protein into the plasmid, and then they form the crack channel, right? So whenever they're like, it spans, the STIM are the protein which spans only one through the membrane, the ER membrane. So they have only the single transmembrane helixes. Those helixes are which span the 10 or 12 amino acid which spans the membranes and protrude onto the cytoplasm. This is the STIM molecule, which the portion of the STIM molecule protein which is bounded by the membrane, the ER membrane, that is called the transmembrane helixes. So whenever there is a store depletion, the both helixes come together and the arm of sitting on the ER from the STIM extend and holds the ORI channel and make tight junctions. So as I have shown on these images are visual representations that although the STIM is distributed all in the ER, and that was the whole ER, but when it gets activated, it forms small junctions. It's not throughout the ER. It's particularly tiny junctions with the plasmid at places. So those transmembrane helixes help two STIM molecules to come together and cluster. It has been reported that it's like 10 to 15 STIM molecules come together to form a single punctum. Single junctions will be formed by 10 to 15 STIM molecules. Exact number are still to be known, and it has been reported like 1445, a very recent paper in the PNS with work of Colin Taylor in UK, Oxford University, I guess, yeah. So it's been reported in 2 months back, something like that. But some of the studies are really diffraction limited. So diffraction limitation is a process in which you cannot see less than the half the wavelength of the light. So what we use a very high super resolution imaging techniques. These are like TIRF, total internal reflection fluorescence microscopy, in which we just ignite with the lasers a very thin section of the ER plasmamembrane junctions and try to localize molecules and their movement. It's like co-localization of molecules with the movement of STIM and ORI. So that was the thing. That's why I tried to show visual representations how the ER plasmamembrane junctions are done.
Thank you, sir. The next question is that what are the instrument techniques which are used in monitoring calcium ions?
So if you want to see the microscopy, you can go for the confocal microscopy. We can go for high resolution microscopy like TIRF I have talked about. If it's not microscopy, it's normally we can also see through the calcium imaging experiments, or we can check the ones with the Western blot, like labeling cells onto the different. So there's like many techniques we can use over there. So I think the method is more important rather than techniques. What you're finding for what is a basic question, then we can differentiate or use those techniques to attain those questions. Electrophysiology is a must for most of the crack channels. You can measure the crack channels, you can measure the inhibitions. That is very important one. And I think Rajesh has shown a lot of data yesterday from the crack channels, and he has done a very nice work. So really liked it.
Thanks, sir. And next, there is one question from Aradhna. Is there any metal that can block calcium channel?
There are various, actually calcium, many channels can be blocked because they're like sodium potassium exchangers and all, and different ones. The lanthanides are very specific. They are like, tri-charge. The lanthanides blocks various channels, but these crack channels are specific for the calcium. So something, I guess, yeah, they can be bound, inhibited by different lanthanides or so, but specific to we are very specific to the calcium channel. So inhibitors must be designed according to the backbone, what we have devised.
Thank you. The next is by Kirti, that what is the role of crack in identifying the molecule as drug candidate, as a potential drug candidate?
So crack is a channel formed by stimenorai at ER plasminogen junctions. So what was the question?
This is how you can, what will be the role of crack in identifying the molecule as drug candidate? Can we use that as a
drug candidate? How, like we use inhibitors to check the protein, right? How we will use protein to check the inhibitors? It's
okay. And one thing, sir, since we are talking a lot of calcium and we're talking of these, so is there any, like they've asked for any symptoms that as an individual one would come to know that you are having a calcium deficiency or something?
Yeah, so no, that's something. The calcium deficiency is a different thing. I was more, yeah, so calcium deficiency is a completely different thing. We can know from our body functions that the immune functions might not be proper with it. Many tests which can show that the calcium is deficient in the body. So you can have the different calciums or the irregular bone development, so muscular dystrophy. But the thing is that the more important stuff to be known that if anybody have a very it's an autosomal recessive disorder. So nobody can know unless you get tested for it, unless you produce your cells or you have to give the cells or you have to check for that conditions. Then you know that if the proper genotypic is done, then somebody knows that it have an autosomal that autosomal recessive disease, and then can be cured with the proper. If you have a very if anybody have a very severe immune deficiency, what happens? So they must be tested for that thing.
Okay.
And calcium inhibition is one of the major subjects right now, and it's utilized in multiple aspects. Nobody have ever thought that Oxora, a calcium crack channel inhibitor, can increase the life expectancy or the ones with the patients with the COVID-19. It was only tested for the acute pancreatitis, right? So that comes with the mechanism. How does the mechanism is devised? How it's like, what is the disease? What is the disease? The mechanism of the disease? What exactly it's causing, and how we can block it.
Okay, that's all for the questions. I hope the questions, the participants, they have got the answers to their questions which they have asked. Thank you once again, Dr. Upendra, for being with us, and it was really heartening to have you here and to enlighten us with your work. Thank you so much.
Thank you, Dr. Manju Mehta. I would really thank the organizing committee this time. Lata Vodwal have worked very hard. She has all the organizing committee as well. So yeah, thanks a lot for inviting me for the talk. Thank you all.
Thank you, sir. It's a pleasure to have you here. Over to you, Lata.
Thank you, sir, to discuss with us the role of calcium release activated calcium channel and its importance in the regularization of the cell functioning. And I believe that our student must get knowledge with the role of these channels in the process of optimization for searching the good drug candidates. Thank you, Manju ma'am, for coordinating this session and sparing your valuable time for the conference. Thank you, Dr. Prajwalit and Dr. Pratibha Chaudhary for organizing this session. And here we conclude this technical session third for the second day of the conference. Now, we will meet again for the next technical session 4 at 1 p.m. Now we have dispersal for parallel paper and e-poster presentation session, and their Google link and the related details are there in conference schedule. Thank you all once again, and I request to join the respective presentation session through Google Meet. Thank you all.
Thank you, Lata.
Thank you, ma'am.